SRA database. Okay. Basically, I am directly adding this in Google. So, what to do from NCBI SRA database? For example, in our last class, selected E. coli just for learning. Why I have selected E. coli? Don't know right now because this is just for learning process. Why we are going to select this data? That is the main question. So for research work, we have to very much clear that why we are selecting a particular organism. You can suggest me any other organism as well. Partha, do you have some suggestion regarding any other organism having less data?
One is you can have Alice types Shahi, Alice types ALI,
Can what?
Alice types. I'll type in the chat box.
Spell please. I'm not
Typing
getting.
it in the chat
Please
box.
type and send. Yes, yes. Okay. For example, we are selecting this organism. This is the particular organism we have targeted. This is the whole genome sequencing. We will not select this. We'll select only Illumina HiSec. This is also Illumina, but maybe there are some. How much data is this? 264 MB. You don't know any other organism with less data?
No, in the list, if you go down, I think maybe you can check.
No, no, no. This data is very much large data, and I don't think so. We can download this data and we'll. Yeah, I
There
think
is
this
one
is
14 MB is there, yeah, 16S. This is
this is 14 MB. Okay, yeah. So we are going to select. This is the basically experiment. We'll copy this experiment, and then we'll click on run. And then go on download first Q file, and we'll download this file experiment here and type, and then we'll select this run, filter data in FASTQ format, and download this data. Okay. Now I have to save this data by the name. I'll rename a folder with this experiment only, SRX2735, and I'll save this data in this particular folder only. So where is the folder? This is the folder. Yeah, data is still downloading. Okay, data is still downloading, and then now data is downloaded. I have to extract the data here. And I'll rename this data A Sahi. Don't know this organism. You have suggested me, so I'll consider this only. Just rename for our own. This is the FASTQ file. Now our first step is to filter this data. I think I have shared a script with you regarding data filtering. Filtering.pl might be. This was the script. I don't remember. I've shared with you. Now, our main focus is to filter the data that we have downloaded. How to filter this data? Using command prompt. Then it's directed towards desktop. CD, where is our data? That is located in this folder. I'll copy from desktop to this folder. We are in this folder. Now how to perl filtering.pl. What was the command? Do you remember? I have shared with you. I'll see. What is the name of input file here? A shahi.first. Read this list of files, and minus f, basically data is from Illumina. Illumina, data is from Illumina only, and layout is single, means this is a single end sequencing, not pair end. So Illumina, and these are the basically some stats. Okay, now see, this is processing. Okay, we have downloaded the filter data. Now filter of data is basically overall the same. Okay, now what to do? Our next step, we have to assemble this data. How to assemble this data? Import Illumina, add files from desktop. Where is the data? This data, this first Q file, open. This is not a paired end read, so we will not go to select this paired end reads. And then next, we have to save the results. Next, we'll save the results in CLC data only. Finish. Now the processing will start here. Data is importing in CLC workbench. Whole data is imported here. I'll close this. Basically, this is the previous file. Now see, this data is here. Okay. Now what will we do? We'll go on toolbox. Here is the option de novo sequencing. where we'll select de novo assembly, and we'll select this data, and next we'll select default 200. Next, by selecting only default things. Next, and we will save our results in CLC data again. Finish. Now work is in progress. Let's see how many reads will matched and how many reads will not match, and how many contigs will be generated, and what is the overall state of the assembly. Okay, we have to wait for some time. That's why I have selected very less data so that assembly will be covered in less time. But whenever you are going to select a data for analysis by means of next generation sequencing, you have to select some data in GBs or in TBs. I don't know how much data will be available for your particular targeted organism. I think data will be more than 1 GB. But if you are going to select some microorganism, it will be around 10 GB. For some plants, it may be more than 100 GB or 200 GB. It depends the organism which you are targeting. Okay. Now assembly is going on. In the meantime, we'll check the reads of this data. How? We'll just visualize this data. What kind of data is present here? So we can see that this is the first read, this is the second read, this is the third read. Almost all reads are same because this is the query coverage basically. Say T, T, T first, then G, then A, G, G, A, A, T. Everything is fine, fine. But in this case, G, G, this is also fine, G, T, C, A, A, T. You can check here. This is G, 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 and again G, C, A. Now, based on per base and based on whole read, we have filtered the data. If if the overall error is more than 75%, then the whole read will be discarded. But if probability score for sequence read is 20% only, means will not accept error more than 20%. And for minimum for each base is only 10%. Means for each base in the whole experiment, we'll select only 10% errors. For each read, it is 20%, and for overall file, it will be 75%. After filtering, next step is assembly. Now assembly is going on. After assembly, what will we get? We'll get some, these all reads will be assembled into consensus sequences known as transcripts or contigs. You can say reads will be merged in the form of will be assembled in the form of transcripts, and we'll select those transcripts in order to find the particular function that we want from that particular organism. For example, if we are going to select some bacteria which is related to uh, particular pathway such as metabolic pathways, so definitely we'll try to find the function of all sequences related to that bacteria, and we'll try to find which protein is related to the particular infection. After selecting that particular protein, we'll go for drug designing. That is just for example, but currently we are just learning that how to download data, how to assemble data, how to filter data. Okay, have to wait here because assembly is going on. Yeah, 99% assembly is done. Yeah. Now see, this is the report for the assembly of this particular data that I have selected. We got 40 contigs and average length was 316 and maximum length is 429 for a single read. And we can see here some stats. 40 contigs were formed. And what we can see here, we have to export this data in the form of FASTA format. I'm going to export FASTA format. FASTA format, select this assembly that we got in this next. This is the folder that I've generated for this particular. Now data has been exported. Now I'll export the assembly report by selecting PDF format. And assembly report related to this organism will be saved in the same folder that we have generated right now. Finished. Okay. Now see, this is the folder that I have generated, the data I have downloaded, the data I have filtered. I'll just remove this data as due to some space issue, might be my computer will collapse. I'll just delete this data. Now this is the assembly file, means transcripts that we have obtained after assembly. See, this is the first config, this is the second transcript, this is the third transcript, and how many transcripts we got? 40. See, 40 transcripts. Now, what is our target? Means what kind of research we are trying to do? Might be in this organism. What is the function of this organism, Partha? Am I audible? Yes, Partha, what is the function of this organism? Means what you have suggested me in this organism? What kind of pathway? What kind of uh, means? What is the role of this organism? Can you please suggest me so that I'll discuss another things
Yeah,
related to that? Yeah, please tell me.
this is one of the gut microbe,
This is a gut microbe. Okay,
and
and its
I'll
main role is in.
I'll just share.
Okay, no problem. I'll just Google this. This is a gut microbe. I don't know because we are just learning. That's why this organism. Gram negative bacteria, anaerobic rod-shaped bacterium from the genus. Okay, no issue. Implication to inflammation, cancer, and mental health. Now, our target is For example, we are targeting inflammation. Now, our main focus will be on which contig is related to inflammation. How will go to identify or recognize that contig, that particular contig which is related to inflammation? So, what we have to do, we'll select first contig and try to find what is the function of this contig. We'll go for NCBI blast, blast X. Always blast X because there are some types of blast I have discussed in my previous classes. And what is the role of blast X? You should please read in either Google, either try to find what is the role of blast X. We'll select only blast X, translated nucleotide, because our sequence is in nucleotide form only, but we are going to find the function in protein blast as well. We'll paste our sequence in blast and we'll go only with non-redundant blast. Means whatever data is available, we'll select that data, particular data and no data. Okay, then we'll go for blast and we'll wait what kind of blast results will come from the first sequence. Then we'll wait for second, third, fourth. There is a shortcut as well. We can select 40 sequences at once for blast, but that we'll discuss later. First of all, we'll try to find the role of this first contig. See, I got the role is this is a hypothetical protein. Basically, this is similar with some hypothetical protein with 97% query, but in all are hypothetical proteins. So this is useless for us. We'll select second, third. I'll select at least 10 sequences at once. Either 5 because 10 will take time. I'll select 5 sequences at once and I'll go for blast. Blast. First we got hypothetical, which is useless for us. What is second protein? What is third? What is fourth or fifth? We'll try to find the gene which we are targeting. For example, gene related to inflammation. What is that gene? For that, we need to go and search some literature for our particular gene. Second query, I'll select and see. This is also a hypothetical protein, no issue. Third, from a single experiment, I'll tell you one thing, 50% data will be hypothetical always. And the information is hidden inside these hypothetical proteins. These all are hypothetical proteins, first, fourth, and what is the fifth? This is also some hypothetical proteins, but we'll try to find some here, some data here. This is PG1 protein homologous to Homo sapiens. Either means these are some results. I don't know whether this gene is related to some inflammation or not. I don't know because these are again hypothetical proteins. I'll select some another protein in this particular data. Because now our main target is to identify the gene related to inflammation. For example, so what will we do? We go for blast, try to find the particular gene. Here I got only 40 protein, 40 transcripts, which is not a big deal to find blast results by using this, but whenever you got, whenever you will get, I think 40,000, 50,000, then it will be difficult for you. In that scenario, you should know how to blast each sequence, and results will automatically come. For that, will I'll conduct another class, we'll discuss later. Okay. Now in this scenario, what is the sixth sequence? This is also hypothetical. 1 2 3 4 5. Okay, second 84, again hypothetical. Don't afraid from these hypothetical proteins, because from hypothetical proteins you can generate a very good information. I have published a lot of papers from hypothetical proteins only. I have selected very less data, due to which I'm getting each and everything is hypothetical in this case. Hypothetical only. No problem. I'll target next 10. Next 5 or 10. I'll select 10. Whenever we'll get the targeted gene from this particular data, then our next step will be to identify its expression analysis or pathway regulation, means which particular pathway our transcript is controlling. Okay, all our hypothetical proteins till date. Let me see it again. What is, there is no problem actually. No. See here. I have found one protein which is PG1 protein. I don't know what is this PG1 protein. I have to find the function of this PG1 protein. Search this PG1 protein. I'll see. Now see. This PG1 protein is basically some sort of polyglucuronase 1. Its role is in carbohydrate metabolic processes, cell wall organizations, and some hydrolase activities, I think. If this protein is related to some inflammation or not, for that we have to search literature only. We'll get results like this only. In this case, I have not got the first hit, but from the third hit, it might be this protein, PG1 protein. BTA 115, but this protein is hypothetical. In this case, another proteins. This is an uncharacterized protein, CAG 260. I don't know what is its role. If this is related to our inflammation target or not, we have to go to literature. After finding our particular targeted gene, we'll go for expression analysis, and expression analysis will be done by using reads data and reference data. Here reference is basically the matched contigs, and read data is basically the raw reads. How to find the differential gene expression by using CLC workbench? I'll try to find here. If not, then I need Linux by using Botai or other softwares which are freely available. Okay. In this case, no sequence has top hit with a particular gene. But third or fourth hit is related to some particular protein. For example, in this case, this is permease activity, maybe some enzymatic activity, I don't know. But whenever you are going to target a particular organism, you would be aware of each and everything so that you can easily select a particular gene from all assembled sequences before finding its expression analysis. Now, till this expression analysis, do you have any query? Please ask. After that, I'll go for expression analysis. If anybody has any kind of query, please.
So even if we take this each of the contig and do ORF, we will be able to find the genes, right? That search also we can do it. ORF that we did initially for each
Uh,
contig.
I'm not getting, please.
We have done that ORF, if you remember, right? So initially, when we have to find the proteins from the sequence data, so each contig, if we search for in the ORF tool, we'll be able to
WARF tool,
find
which tool? WARF means
ORF.
ORF.
Yeah.
No, 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 no. Actually, that is a separate thing. I'll discuss that later. ORF is basically just for if I'm not able to find any protein from these transcripts, it means there is no translated region. That's why we are not able to get some protein.
Okay.
Okay.
So it's not like we are open reading frame. If we are able to find in these contigs, that doesn't mean we have a protein, is it?
No, no, no. Because currently we are not able to find any particular function. If there is no particular function and we are going to perform blast X means, but one more thing I'll share. This is the NCBI blast. I just go back. Might be I'll get some results by using blast N because this results is with blast X means translated proteins only. Because I am targeting a particular protein database so that we'll get some protein and we'll check the expression analysis of genes. And after that expression analysis, we'll be able to find the particular gene, which is now I'll select blast N. This is with nucleotide sequences only. And I'll paste my sequences here. I'll get some with nucleotides only because previously I have targeted proteins only because each and every protein is related. For every disease, protein is a main thing. So I was focusing on protein only. But for a particular function at nucleotide level, we can find. Okay. But what you are, I know you are saying that we'll try to find ORF, means we'll translate our sequences. Tool, Expressy Translate, and I'll try to find the largest sequence, for example, the 17 that I got hypothetical. You see, the translated region is only this much. Might be there is no function of this particular translated region. And in blast X, nucleotide translated to proteins and then it will blast with protein database. Might be this is the reason we are not getting any particular function and this comes under only hypothetical proteins. Okay, but we can select the particular translated region, but this is very small region. So I don't know what kind of protein is this. If we are targeting some small proteins like insulin, we have discussed previously, then we can check by performing ORF and we'll check what is the function of this particular ORF despite of its length. Okay. Yeah. Any other query? Swanti,
Yes, sir.
I don't know when you have joined. You have any query?
No, sir.
Okay. And Yash,
Sir, what if we when we do the blast, then you get the hypothetical proteins, similar some similarity to the hypothetical proteins. So hypothetical proteins, what are they? So do they have any functions regarding to it, or we have to just find the protein that is important to us?
basically in my previous lectures, I have discussed a lot about these hypothetical proteins, but I'll repeat it. Hypothetical proteins from every newly generated data will get, now see here, NR blast with nucleotides. There is no hypothetical protein from it is resembles with some uncultured bacterium in NR blast with nucleotides only. But I'll first of all I'll clear your doubt. Basically, whenever you are going to generate transcriptomics or genomics data related to a new organism, you will get 50% of the data is hypothetical. Means there is information in hypothetical proteins, and these are really very important information which is hidden in the form of hypothetical proteins. But we are unable to find or extract the particular information from hypothetical proteins, for which we need to conduct a separate research. Okay, I'll share my some of my publications with you where I have focused only on hypothetical proteins in which some important information is hidden. I have tried to extract that particular information which is present in hypothetical category. Means everybody is targeting only the information which is available and they are ignoring hypothetical part. What I have done, I have tried to find hidden information and uh, available information as well. And I have reported this work in some research papers. Might be Dr. Renu will share with you. I'll open a publication related to hypothetical proteins. Means hypothetical proteins can be downloaded, can be targeted for finding a new information. Okay. And these are really very important proteins for us, and they are present in our genome, in each and every organism's genome. And this information is also transferring from father parents to offsprings. Means hypothetical proteins are basically they have phylogenetic lineage as well, they have important functions, they have hidden transcripts as well in hypothetical proteins. Now I'll share. See, this is a publication where I have selected hypothetical protein, uncharacterized proteins. Basically these are hypothetical proteins. I have tried to find their function in particular organism. This was the organism that I have selected, and I have characterized these hypothetical proteins by proposing some particular methodology. See, this study reports structural modeling, molecular dynamics of profiling of hypothetical proteins from this genome. And what was their role? I have tried to propose some means what was the conclusion of this study? Proteins are envisaged as possible targets for developing drugs for this particular infection, and these proteins were present in hypothetical category. Now this is the basically separate part of the research, despite from NGS is basically when we are going to find the particular gene from our assembled data. Now what is the expression analysis of this particular transcript? First of all, assembling the data. Second, first of all, selecting the data. I have selected only single run. I have to select more than 15 runs, 20 runs. and I'll download those runs, and I'll filter my data, and then I'll assemble my data. After that, I'll go for functional annotation of that data, where I have find functional annotation with NR blast, but with nucleotide databases. I have found some infection. Now I can say this is the amplified from, I don't know, this is the first gene which is I think not relevant with our with this particular study. Might be second one will be relevant, might be third one will be the relevant. Uncultured ribosome. Now see, might be 16S ribosomal RNA gene. What is its role? I don't know what is its role. Might be it is important for the study regarding this organism. Now we'll select this particular gene only, and we'll try to find the expression analysis of this gene. How to find the expression analysis? We'll go in CLC again. I'll try first. I don't know CLC will work or not. Toolbox, map reads to contigs. Here I'll select only map reads to contigs. From each read, we can say that how many times this read is basically present. Okay, no asking. Now I'll select contigs. Where are those contigs? Okay, this is not performing actually. I have to download some contigs from this reference data. I'll try to download. No issue. This is not working here. Might be mask. I'll do this work by using Linux only. This is not working here. Actually, we have to map some contigs. We have to back map some reads with contigs, and we'll check how many reads are mapping with a particular contig, and for that particular contig, what is its expression analysis in comparison with the diseased and normal sample, and we can easily say that expression analysis of a particular gene is regulated, might be upregulated or down regulated. I'll again suggest to you, I'll again discuss this thing because this is really a difficult thing to understand. Now, for example, in this case, contig number 17 means transcript number 17 was very important for our studies. For example, okay. So its role is in inflammation. Now I have to check whether this transcript is upregulating or down-regulating. Then how will I check the expression analysis? What I can do, I'll map my reads with that contig, that how many reads are mapping in a particular contig. The expression is basically reads while generating data. I'll map those reads with a disease condition and normal condition. Then I can get for contig number 17 in disease conditions how many reads are mapping. For example, in disease condition 20 reads were mapped, and in normal condition 40 reads were mapped on the same contig, on the same gene. Means in disease condition only 20 times and in normal condition 40 times. It means in disease condition this particular gene is down regulating. How much down regulating? By half. 40 by 20 is 20. Basically 20 by 40 is 1 by 2, and that is 2 by 1. Okay, that is down regulating by half. And up regulating, if we'll see up regulating, then it is upregulate. If in normal condition it is 20 and in disease conditions it is 40, then in disease condition it is upregulating by 2 times. 20 into 2 is 40. Okay, we have to map reads in reads related to normal condition and reads related to disease conditions. We'll download that data and we'll map those reads from normal and disease condition to our assembled data. Now, if you have any doubt, please ask me because I'm trying hard.
Sir, but the read is not a gene, right?
Read is not a gene, but read is basically it is presenting in a particular NGS sequencing time. Means read is there, it means this read is expressing.
Oh, but there will be a less probability, right? Probably even if read doesn't express, some part of that read might be expressing which we might miss out, right? Or
No, 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 no. While mapping our reads, again we'll select a probability of 75% error. Means
oh,
75% read should be mapped.
okay,
Okay. When 75% read should be mapped, because then we can say 10% read is mapped, we can't count in as a one expression. We should count whenever 75% read will would be met. Means this error will vary from 63 to 100. Up to 63% we can consider a read mapping, because might be due to some errors of bases, either due to some errors of some 3 dash or 5 dash or intronic regions. So we can consider from 63%, because I have considered 63% in one of my research publication. Okay, but not below 63%.
okay,
Okay, at least 63% read will be mapped, then we can consider that read is mapped. But if it is less than 63, then that read should be excluded. Okay, for read mapping I need a Linux system where we'll do some mapping work. How to do, I'll try to manage that work by using Linux system. For read mapping, we need a tool known as Bowtie. I'll text the name Bowtie 2. This is the tool by which is freely available, but it will work only on Linux system or Mac system, where it will map the reads with contigs and reads from disease condition and reads from normal condition. Will map those reads to the assembled gene or assembled transcript that we got after blast analysis. And then we'll compare how many reads in this particular gene are mapped from disease condition and how many reads from normal condition. And then we'll compare both and we can say what is the regulation of this particular transcript during disease and normal condition, and which pathway this particular transcript is targeting, and what might be done in order to cure this disease. Might be we have to overexpress that gene, either we have to down regulate that gene, either what is the protein product of that gene, we have to target some drug for that protein product. Means what we have to do, we'll get the details once our expression analysis will be completed.
So all the experiments have control and disease experiments SRR value. If
Yes, yes.
experiment on okay.
They should have, because if they don't have any control or disease, then they can't be able to, then they are just generating data that this is data related to this particular organism. Why you are generating this data, they don't have answer, because in all my publications I have generated data related to disease and normal condition. I can show you as well in my publication. I'll show you, because this is comparative analysis. Mm, This is basically related to blister blight. We have, first of all, we have targeted I published two papers related to blister blight. One is transcriptomic analysis and other is from hypothetical proteins. Now I'll directly come to methods where I can say that one is resistant plant. Resistant plant was basically the plant which is resistant to blister blight disease. The blister blight disease is basically a disease caused by fungus E. vagans, and in the same location one plant is resistant to this fungus and other plant is susceptible, means this plant is affected and this is not, and both are the varieties of camellia sinensis. Just see what was the gene which is in resistant variety and which gene is not regulating in susceptible variety, so that this thing has been happen in this variety. I This was the basically idea. Then what we have done, there is a heat map. I'll show you in results. Here what we have done, this is the resistant variety, this is the susceptible variety. Both are T varieties. We have selected sample from resistant and susceptible, and then we have mapped resistant versus susceptible. How? I'll show you in a heat map. Means what is the expression? This is the assembly basically. Yeah, see this. We have found 149 immune related genes from resistant genotype and susceptible genotype. Now see, I have managed RG means resistant, SG means susceptible. This particular gene, disease resistant protein, it was highly it is down regulating in susceptible varieties, but it is upregulated in this resistant varieties, and in some other genes which are related to, they are all, but we have to find which genes. See, for example, this first gene named as disease resistant protein RPS2. This is not expressing in as susceptible varieties, but it is overexpressing in resistant genotypes. So we can easily say this gene is very important for the defense of blister blight mechanism. Blister blight defense mechanism, we can easily say. So from all those contigs, we should analyze in this way only, by comparing no control and infected samples, and then we can say, we can propose the basically how many contigs or how many immune related genes or how many other genes. We can, whatever our idea is, we can easily say this is the particular gene which is related to this particular thing, which is expressing down or up in disease or in normal condition, and we have to dissect this gene by using some conventional wet lab methods. But in my case, we are saying that if we will upregulate our this disease resistant gene in susceptible variety, production of T will be high, which will be beneficial for our country. But in case of diseases, we'll say if we can reduce the expression of analysis of this gene, either we'll try to find some drug for the protein product of this gene, might be disease can be cured. So we should target always so that controlled and experimental other data which is related to some particular disease will be available, because we are here, we are going to dissect only by using in silico means. We are not able to generate data right now, but if you have facilities to generate the data, then you can select the single disease data, and by means of real time PCR, you can say this gene is overexpressing by using real time PCR, and then you can analyze that that how many reads are there and which kind of protein product is there and what is the structure of that protein, you can also analyze by generating the data. Okay.
Yes.
Any other query?
So now for this particular example, we got 40 contigs right now, sir. So can these 40 contigs be treated as hypothetical proteins?
No, 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 no. We can't treat this because this is just for learning process. I have randomly selected a single sample, and how many samples are there? Even I don't know. Maybe there are more than 10 samples in particular experiment. Might be more than 10. This is just for learning. We should select if we are targeting this particular organism, then we have to download all the samples related to these organisms related to one particular condition, then we'll assemble, and then if we got some hypothetical proteins, then we have to dissect those hypothetical proteins. Okay, because this is a just one experiment, one study, and how many studies are available, might be there are some errors. I don't know about these studies. What is this? If you are targeting a particular organism, how much data is available, that should be the first point you have to focus. And after downloading whole data, then you can assemble, might be, not might be, I'm sure 50% protein of the data should be in hypothetical category, and then you can target that 50% data, and you should try to characterize that hypothetical part in order to find some hidden information. In my case as well, where I was trying to find these immune related genes, 50% data was hypothetical. That's why I have targeted another publication. So I was successful in identifying some new information from that hypothetical part by using my hypothetical methodology. In the end, I have done some wet lab work as well, but that was not so much relevant. That was only real-time PCR analysis, nothing else. I've done here. I have selected same data, but only targeted hypothetical proteins, a characterization of hypothetical proteins involved in blister blight, and the from our previous study only. Means C, biotropic fungus responsible for this, and these proteins are categorized as hypothetical proteins. And I have downloaded this same work, and I have found how many proteins were related to blister blight disease in this hypothetical category. I have written, I think, see, 36 hypothetical proteins upregulated in tolerant genotype. I have found some genes in hypothetical category as well, and I have proposed that these genes can also be used for dissecting blister blight disease. Okay, this is actually a paid article right now. I am not able to download this article. Okay, you can go through these two articles, that how many proteins. Yeah, see here. Of these 213 hypothetical proteins, just imagine, from hypothetical we got 213 hypothetical proteins, which has some noble putative defense relative function categories. And from the main publication, how many were? 149. If I remember, that is 149p. Means from hypothetical category, we have found more than the normal one, because at that time T genome was, yeah, 149 here. And in hypothetical category, how many? 213 hypothetical proteins. Just think. This is the difference. But at that time, in 2019, either in 2016, whenever we have done this experiment, this work, this is published in 2019, but I have done this work in 2018 before that, because this work was published in 2016 Yeah, 2016. At that time, T genome was not available, but in the current scenario, T genome is available. So at that time, I have gone through de novo assembly, but right now we have to select reference based assembly. In reference based assembly, hypothetical proteins will be less than original annotated proteins. Okay. Why hypothetical proteins are more? If we are going with de novo assembly, means no reference is available, then hypothetical proteins will be more. Okay. But if fully annotated genome is available, then transcriptomics data, hypothetical proteins will be less. But they are hypothetical proteins will be there, but in less amount only. Not in less doesn't mean 10%, 20%, might be 40% or 30%. This less means less than 50%.
So in your study, you went with de novo, right, now, sir? So that means that you will be setting the reference for your
No,
organism.
no, right now, I don't know about this organism. I don't know genome is available. If genome is available, where is that genome? Then I have to download that genome and I have to provide reference and then I have to map these reads to that reference in order to assemble. But for de novo assembly in transcriptomics case, you can easily do de novo assembly in order to find the particular gene in order to compare, only for comparative analysis.
So de novo is much less accurate than reference-based
No, no, no.
assembly.
Both are fine. But genome assembly is basically reference-based assembly has some more benefits because if genome is available, lot of people they have done work on genome for a particular organism. Might be more annotations are available. So there are less chances of hypothetical proteins or uncharacterized proteins. But if no reference is available, then you have no other option. Just go for de novo assembly. Hypothetical data is available. Just dissect that hypothetical data. Either try to find some other kind of data from RNA-seq analysis only. Okay. I think this is really a very big thing to understand expression analysis, to understand assembly, and I have tried very hard to in order to imagine you how these things will happen. If you have any other query, please. So now I am giving you a assignment. I have already discussed with Partha and Swanti that you should generate a idea that which kind of data and why you are selecting that data and which kind of data you have to select and download from NCBI SRA database. That assignment is really important because by some idea you will start reading some literature related to organisms or diseases and you can easily find that how much data is available and what we have to do now. If genome is available, then reference based assembly. If not, genome is not available, then de novo assembly, and then identifications of functions. After that, I'll suggest you how to find other things like how to find pathways, how to find uh, but other things I think Partha and Swati has done already, that how to find the localization and physiochemical characterization of these proteins, and then structure analysis we have done in our previous lectures. But for NGS, assembly, annotation, and then differential gene expression, means that is a expression analysis, and after that we'll do some sort of protein-protein interaction network analysis. But before that, we should know how to find pathways of these particular genes, which we have extracted from next generation sequencing data. Okay. Our next target is to find the pathway of a particular data. Then we can easily say that this gene is really very important. This is targeting a particular pathway. This is really overexpressing or down regulating. We can easily say, and we can publish this work up to structural biology, up to interaction, up to some other parts. We can propose some drugs for that particular protein as well. Okay. I think we should stop here. If you have any query, please ask me.
So, how will the expression look like after we map with the context, like the heat map which you showed?
Yes, yes. There is a
Mm.
software which is known as TMEV. Basically, that is TMEV is a software, TMEV, that is for expression analysis. It will give you results in the form of heat map. Okay, heat map, but the expression will come in the form of only values. Like for, I'll show you in one of my presentation how expression analysis will come in the form of some values and how we have to calculate that expression analysis. As I have done this work, so it's really very simple for me. But for beginners, this work is really a very big deal. Means how to select data, how to analyze the data, and what kind of things you need for your research work. That is really important. This is the part that I have shown you: quality control and filtering, then de novo assembly, then assembly softwares, then annotation, in which we have got some blast, and then I'll show you geo analysis, AC number and KEGG pathways analysis. I'll show in my next lectures. This is basically NGS, where Illumina sequencing base culling. When we are generating the data, this is FASTQ format that I have downloaded, and the same thing SRA, download our data. Now it might be easy for you. After assembly, this kind of data will get, and this is the context stats that we have get. This is the basically annotation file. Okay, I have not written here expression part. I'll open expression part in my another presentation. This is just annotation part. This is almost similar with those things. This is basically reads. These are assembling the reads and generating. This is the annotation. This is the pathway. For example, I have selected here glycolysis pathway. In glycolysis, these green are our genes that we have found in the particular pathway. And this is the basically expression analysis. For example, contig 1 in first condition by second condition, second by first. These are the basically values. For example, how many reads are in normal and controlled, and what is the function we have written here. And these I have highlighted red, means this is over expressing in one of the particular condition. Means contig 1 or transcript 1, and the values 1 by 2 or 2 by 1. Okay, this is just in the form of values. And for visualization, we'll go for heat map. Okay, and after that, after identification of these contigs, we can also propose that what kind of markers are present in this particular gene, so that for each and every time. We don't need to analyze in the form of next generation sequencing. By mapping markers, we can easily find that this gene is available here or not in other plant or other animal. We can easily find some single sequence repeats, single nucleotide polymorphisms, intron length polymorphisms by using this NGS data. And finally, by using some primer synthesis, we can validate our sequences in wet lab as well. And we can also find the enrichment analysis of our particular genes, means this is upregulating, down regulating, either is a positive regulator, negative regulator, or only regulate in simple way, and in which particular pathway our gene is basically working, so that we can select this gene or protein for our drug designing project or not. Because if we are randomly selecting any protein for drug designing project, might be that is more dangerous than that disease. Might it will finish our time in might be it is more poisonous than the particular disease, because function of a protein will be stopped, then maybe some important pathways will be regulated. So we should be careful before proposing any particular target related to humans. But for microorganisms, we can easily propose a drug designing target, so that the pathway of a particular microorganism can be stopped, and that microbe can be finished by using the particular drug. Okay, after checking the toxicity of that drug. Okay.
Okay, sir.
Yeah, I think we should stop here. Renu ma'am, do you have any query?
You are already speaking since a long time. I can ask my query in the next class, maybe.
No problem, ma'am. No problem. You can ask, because this is really very good flow.
Yes, I had something in mind, but it slipped out.
Oh.
So if I remember, I'll ask. Yeah. Anyone else has any doubt can ask maybe.
Partha, you are not audible. I think you want to ask something.
No, no, sir. Right now, no questions. If I have anything, I'll ask.
Okay, okay. Yash.
No, sir. All good.